Yes. So, as I mentioned you, so I'll give a very high level uh, explanation of the mechanism. Uh, in bacterial system, there are three important components. One is uh, Cas9. It is an enzyme. So think of it as a molecular scissors, a scissors which can cleave any foreign DNA. So Cas9 is a nuclease enzyme acting as molecular scissors, and along with Cas9, uh, there will be some other elements called cRNA or track cRNA within the bacteria, which help to identify any foreign DNA. And then when it identifies the foreign DNA and binds to the foreign DNA, the Cas9, which is a nuclease, which is a molecular scissor, will cleave that foreign DNA. So that is a broad overview of mechanism. But when it is re-engineered and being applied to human therapeutics, here the key component is one guide RNA. So guide RNA is nothing but an RNA which guides Cas9 to a specific site to degrade the genome. You can think of it as a molecular GPS. So guide RNA could be a molecular GPS kind of thing, and guide RNA is a synthetic RNA which can be synthesized in the lab based on the specific target sequence that we need to target and clean. And guide RNA along with Cas9 enzyme, Cas9 protein will form a complex, a ribonucleoprotein complex, and it will bind to target DNA at a place called PAM, protospacer adjacent motif. So, uh, and, in the upstream of 3 to 4 cases, typically when in a genome, when there is NGG sequence, generally that is called a PAM motif sequence, and there this CRISPR-Cas9 complex binds to those at that specific sequence, and then it helps cleave the DNA. And this CRISPR-Cas9 complex help in inducing double-stranded DNA break, approximately 3 to 4 nucleotides upstream of PAM set. And when the DNA is cleaved, cell will automatically have a repair mechanism to repair the cleaved DNA, and the repair will be done through two modes. One is non-homologous end joining, and second homology directed repair. In non-homologous end joining, there will be a gene disruption because it will not join uh, correctly, and there are errors, and it will lead to some insertions or deletions, and finally some mutations. Whereas homology directed repair, a target gene sequence can be inserted during the repair so that the genome will have the new target gene sequence of our interest. So basically, within a cell, within a nucleus, we have genome, and within the genome, at a specific point in the genome, uh, we are the CRISPR-Cas9 will bind to that specific point, cleave the DNA, and either it can insert mutation, so there will be a gene loss of function of the gene, or if we insert a target DNA, then the target DNA is inserted at that specific location of the genome. So CRISPR-Cas will act as a scissors. It will cut the DNA, and at the site where it cuts the DNA, it will introduce a new gene of interest. Looks simple, but a very complex mechanism. This is done within cell, within nucleus, in the genome, at specific locus of the genome, at a specific palindromic sequence. The DNA is cut and then rejoined with a new target gene or get a gene sequence to get a new function, gain of function or loss of function of the gene. So this is broadly a mechanism of CRISPR-Cas9, and researchers have been developing different variants of Cas9. Cas9 is a nucleus, acts as a molecular scissors. It will cut double-stranded DNA. There are improvisions in Cas9. There are Cas12, Cas13. Some will cut RNA, some will cut single-stranded DNA, some will only help in inducing one base one base pair editing or one base pair change. So there are several versions of Cas9 which researchers have developed and are being used in different types of gene editing tools. So finally, CRISPR-Cas9 is a third generation gene editing tool, an advanced gene editing tool. The first generation being zinc finger nucleotide, second generation being talents, and this is the latest third generation gene editing tool, which is most used because of high sensitive, high specific nature of the CRISPR-Cas9 tool. Yeah, as I explained, there are different modalities, means different uh, types of CRISPR uh, mechanisms based on the Cas, based on the Cas enzymes. And Cas9 is most common used. Cas12 it is used for some diagnostic. Cas13 as well in diagnostics. And there are different other base editing only to change one base pair within the gene rather than changing a complete gene or changing a fragment of the DNA sequence.